Hi class. Alright, so mitosis in 5 minutes. So we have a cell, right? And the cell needs to go through the division, right? It needs to divide. So it needs to replicate itself. So what do we have in cell? In cell we have chromosomes. So there are 23 chromosomes from mother, and there are 23 chromosomes that comes from father. So altogether it comes to 46 chromosomes. So each line represents 23. So this is the karyotype. It's a display of the pairs of chromosome in a cell. So you see here, that's chromosome, let's say, from father, that's chromosome from mother. So, but each chromosome represents 23 of this, 23 of that. So this belongs to father, this belongs to mother. Right? So it's 23 pairs. 23 pairs. 23 pairs gives you 46. So 23, including sex chromosome. So that's why, for the sake of the paper, so we're not gonna go like that, like 23 times like this, right, for the mother, and let's say 23 4 times for the father. So that's why one line represents 23 chromosomes. I know that's where the confusion comes in, because some people cannot like visualize that. So before cell divides, it needs to replicate itself. So what happens, this chromosome, I'm gonna use like that. So let's say this is original. So now, for the proper division, now we have to make a copy. And we make exact replica. So now we call them sister chromatids. Same thing, so it's gonna be 23 of this and 23 of that. So the same thing happens with a mother's chromosome. So let's say that's the original mother's chromosome, and then you make another copy, so it's 23 and 23. So you have 46. And here 23 and 23, you have 46. 23 old, 23 new, doesn't matter, they are identical, exact replica, sister chromatids. So now we have to properly divide them. So now, in the cell, through the metaphase, right, there's like stages that we'll discuss in class. So in metaphase, this chromosomes, they align, let's say, like vertically, one on top of the other. So what will happen during mitosis, these chromosomes will align this way. So let's say this sister chromatids, two exact replica of one another. Let's say this one belongs to mother, this one belongs to father. So we need to separate them. We need to split them in anaphase. So the anaphase, that's the stage when they will be pulled one from another. So basically, vertically, we align them, and it doesn't matter in what sequence. So for example, this belongs to mother, right? And this 3, let's say, represents all 23 chromosomes. And everything that is in blue will represent also all 23 chromosomes that belong to father, right? 23. So in the next stage, in anaphase, they will be pulled to their respective side. So this red, let's say mother, right, will be pulled here, pulled here, pulled here. So you will pull 23 that belongs to mother. And also to that side, to the same side, you will pull 23 that belongs to father. And that gives you 46. So it's a complete cell, new cell. Similar will happen on the left side. Go here, here, and here. Same thing, 23, this 3 represents 23. And the similar thing will happen with red, right, with mothers. Also 23. 23 plus 23, 46. And it's just another cell. So we do not add anything. To get to this point, that's what we have to do. So there's a copy of one chromosome, copy of another chromosome for mothers, and copy of fathers. So, and if we're going to look at karyotypes, here are 23 from each parent, right? So it's the 23 pairs that gives us 46. So this blue represents 23, and this red or green, whatever, represents 23. So the blue's gonna be pulled on one side and the green's gonna be pulled on one side. Same thing is here, 23 and 23. So one cell will end up with 46 chromosomes and another cell will end up with 46 chromosomes. So let's say this cell will be a new stem cell, and this cell would be a new specialized cell, like if it's a skin cell, for example. And we'll continue dividing into skin cells to replace the old skin cells. So the mitosis, it is a type of cell division that results in two daughter cells. That's what it is. Let's say this is the mother cell, not the mother that we were discussing, mother and father. It's just the cell that gives birth, let's say stem cell. So it ends up with two daughter cells. That's why we call them daughter cells, because it starts with mother cell. So the daughter cell each have the same number and kind of chromosomes as the parent nucleus. So when the cell has mother's and father's chromosomes, we call them diploid, 2N. Have two copies of the genome. So the genome, it's your entire genetic makeup. That's what you are. From the father, from the mother, 23, 23, 46. Identical to parent cells. So the mitosis is just a cell replication, so we can proliferate, so we can replace old worn out cells, repair of damaged tissues, replacement of cells. So there's like four major stages. One is the prophase, then there's a metaphase, then there's anaphase, and then there's a telophase. So the prophase, before or earlier. So before the cell division, chromosomes condense and become visible, right? Visible. Before that is just like chromatids, like bunch of noodles. We cannot see them under the microscope. But in the prophase, they start condense in preparation for division. So that's why we can see them. When you already see it this way, chromosomes, it means that it's already a sister chromatid. It means that they already replicated their original DNA. One from father and one from mother. So that's the new, that's the new. So the second major stage or phase is the metaphase. That's when the chromosomes align on this metaplate. Again, 1, 2, 3, 4 represents 23 that belongs to mother, 23 that belongs to father. So just because, let's say, this is the original and this is the copy, it's the same thing. Yes, from father you want to bring 23 here and 23 here, and let's say this is the mother's, 23 on this side and 23 on that side. So the chromosomes, right, they are attached by the spindles that will pull chromosomes to their respective poles. So the anaphase, it is backing or against or away. So then you just pull them. So it's 23 mothers and 23 fathers. 23 mothers and 23 fathers. You end up with 46, you end up with 46. That's under the microscope. So if you're looking at the cell under the microscope and you see this, that's anaphase. That's when the sister chromatids being pulled to their respective sides. Under the microscope, when you look at this, you see they are aligned on the metaplate, or metaphase we call it, right? The metaplate. So then you know that this is the stage of metaphase. So then, when one side has 46 and another has 46, and then in cytokinesis, that's the division. Well, it actually occurs after the nuclear division is complete. So this creates a cleavage furrow, and that separates the two daughter cells that will have 46 and 46. And you do not see chromosomes anymore because they are not condensed anymore. So again, they look like that. Again, like 23 from father and 23 from mother. So this basically like major steps on mitosis. So there's a lot of little things, but for our class, I just want you to have that general perception on what mitosis is. So basically we didn't mix any genetic materials, we just doubled them, we just copied them, so we can create two daughter cells. So one could be a stem cell, for example, another could be a specialized cell that will be producing new skin cells, for example. So you can look under the microscope in a slide, and there's like a bunch of cells. You see like one cell, there's another cell. Not necessarily that all of the cells that part of this tissue will go simultaneously through all of the steps. Because let's say this cell, you see this cell goes through the metaphase, but the neighboring cell goes through anaphase. Let's say this cell do not go through the division, or they're prepared for the division. This one goes through the telophase, this goes through the prophase. Obviously you have to have a trained eye in order to tell the difference between one cell and another and what is going on in that cell. But it's not the purpose of this class. So that's basically a simple overview of cell division. So, and I do recommend to do that. You have a cell, mother chromosome, father chromosome, 23, 23. So before the division, there's a copy, sister chromatid, on mother sides, and there is a copy on father sides. Then they will be aligned in this way on the metaplate. So that belongs to father, that belongs to mother, 23, 23, or 23 mother, 23 father. I'm sorry, 23. So 23 for father would be pulled on this side, and 23 for mother would be pulled on this side. And on the other side, same thing. 46, 46. I hope you get it.